Hi guys, we're happy to be here with you today. So we're going to show you protein synthesis. First we're going to start off with transcription.
Well, first we'll just tell you what our foods are.
We should tell you that.
So the blue cupcakes are the nucleotides for DNA. The yellow are the nucleotides for mRNA, and the pink are the nucleotides for tRNA. And then the red, orange, and yellow Twizzlers are covalent bonds. The blue and purple Twizzlers are hydrogen bonds.
The brownies represent the amino acids for the tRNA, which we'll get to later. Okay.
And then that green.
And the green is a peptide bond.
Just so you know.
And a nucleotide, if you didn't know, includes the base, the sugar, and the phosphate group. So let's get started. All right, so first we have our DNA right here, all connected and wonderful. And so we take our RNA polymerase, which initiates transcription, but first it has to bind to the promoter, which is a sequence of nucleotides that begins transcription. So the polymerase would bind, let's say, here. And once it binds to the sequence, then these bonds are taken out and the DNA separates. So we're going to take this strand away. And it goes bye-bye and leaves.
Yeah.
And so and so then here comes our RNA, and so one strand at a time, the corresponding nucleotide comes in and binds with the DNA.
Through a hydrogen bond.
Yeah, and then the covalent bonds come in later since they're free floating. So we'd add in bonds here. There comes the bonds.
So then these are all going to bond.
Mm-hmm. So when the RNA goes down, and that's how each one bonds with the hydrogen bond, so it keeps going down, sort of like a little elevator, and then once it reaches the termination signal, it stops, because the termination signal is basically like the opposite of a promoter. It tells it to stop and it ends, and so then once it reaches the termination signal, these bonds are yet again broken, and the two strands separate, and the RNA polymerase leaves and starts this all over again with another strand of DNA. So that's transcription.
Yep, so then we're going to move on to translation. So this strand of RNA is going to move out of the nuclear envelope, and then this is just another, it's the same thing here. So then a ribosome is going to initiate translation. So we have our start codon, which is AUG, and then this is our tRNA, and this is the anticodon, and this is methionine. I think that's how you pronounce it. And so then this tRNA is going to come in, and it's going to form these bonds, and then we're going to have another tRNA come in right next to it, and then form these bonds, and then and then this is going to, these peptide bonds actually, well, this is one. So that's going to form, and then the rest of this tRNA is going to separate and leave.
Yeah, these cupcakes are just going to go away.
So then we have, actually we have it right here, and then this is, this is our stop codon. So this signals that no other
no tRNA can bind with that one?
Yeah, no tRNA can bind. There's no code for that. So then this is removed, these bonds are removed, and then you have your polypeptide chain here.
And this is our resulting one right there.